From Microbe TV, this is TWiP, This Week in Parasitism, episode 256, recorded on March 28th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about parasitism. Joining me today from New York, Daniel Griffin.
Actually, Vincent, I'm in Greenland. I'm only joking. I came over. I thought a whole bunch of other people were coming. Apparently, no one showed up. Just me. No, I'm here in New York.
Your bow tie is totally black, isn't it? There's nothing on it.
No. It's one of those things like the mirror. It says the quality of the recording that will be recorded will be higher than that which you're seeing.
I see.
For those on YouTube who can look closely, this is a really nice, well-tied navy blue bow tie with these red ribbons. And it is an HIV awareness bow tie.
HIV? Yeah, so it's an STD, so it's Friday.
On Friday, you got it. Yeah. And I think, if anything, right, this is going to be a major issue going forward because of all the withdrawal, just the thousands of new diagnoses, over 1 million excess deaths we're forecasting with some of the recent changes. So, but I will let you introduce our other host before we go down that rabbit hole.
Also joining us from Glasgow, Scotland, Christina Nowla.
Hello, good evening, everybody. From a not so sunny Scotland, but otherwise it's been a very good week. So,
you guys think we're crazy here in the US, probably, right?
Well, no, because that would not be really fair. I mean, some people are crazy, but I think a lot of people are not. It's just a little bit puzzling, worrying.
Yeah,
I don't know how to express it, but it's not
the power of misinformation.
Yes. Absolutely.
I don't think this is what a lot of those folks signed up for. So
no. But you know what? Just briefly about viruses. I recently listened to TWiP, the podcast.
Yeah.
And they were talking about measles. And actually, what I wasn't aware of, and I checked it up. Well, in Europe, we've had a lot more measles cases than you have in the US.
Yeah.
But we don't really hear about it here in Europe, so I think I'm going to start raising awareness. And I've made a call with my pharmacist to have my MMR because I've never had one.
Really?
Oh my gosh, Christina, I didn't know you were anti-vax. What's happening?
No, I'm just old.
When did you?
I had a measles vaccine and a rubella at a later stage. So I think in 1972 I was vaccinated against measles.
Were you you never had measles, as far as you know?
No, I never had measles.
Well, you better get vaccinated.
Yes, yeah. I've been speaking to my pharmacist, so
to raise a little measles awareness. So 1971 is when the MMR came out. And you all in Europe had over 100,000 measles cases last year.
Yes. Yep.
You're there not far north of the birthplace of the anti-vax autism misinformation. So.
Oh, that loony bin, yeah. Sorry, that was rude.
He's a loony bin.
Yeah.
Yep. Well, if you like these discussions, we'd love to have your support so we can continue them. You can go to microbe.tv/contribute for different ways that you can help us out. Before we get into our papers, I noticed something in morbidity and mortality this week, which I just wanted to mention. It's a fatal case of splash pad associated Naegleria fowleri meningoencephalitis in Arkansas. So, we've talked on TWiP about brain-eating amoeba. You jump into a pool or a lake or a stream, and the water shoots up your nose, right? And it gets the amoeba get in your brain. But this is a splash pad. I had no idea that you could get it from a splash pad.
What is a splash pad? Sorry, apologies for my ignorance, but is that a kiddie pool or something?
Yeah, it's like it's a area where it's padded, and then there are various fountains that spray water up, and the water tends to accumulate on the pads, and
and you jump and have fun.
You jump around, and it can go in your nose, I guess, right there.
Yeah, it could shoot right up the nose, go right through the cribriform plate, and some of these warmer situations that we are down in Arkansas, the amoeba can end up in the brain, and
So they say this wasn't properly chlorinated. That was part of the problem, because in a lake you don't have the ability to chlorinate, obviously, but in these artificial situations, it should be properly chlorinated.
Yeah, it's interesting. I wonder, because you think they're using, treated water, but I don't know. Yeah, no, it's horrible. I'd seen this. I haven't gone through and read it. I figure this will be something I'll discuss on the Infectious Disease Puscast. So.
They say in the U.S. from 62 to 2023, 164 people have acquired this, and only 4 have survived. Isn't that horrible?
Really horrible.
Anyway, this was I think it was a single case. What do we have?
Yeah, and just the MMWR, right? Not to confuse that with the vaccine, but that's okay if you do. The MMWR right before was a case of a returning traveler with sleeping sickness. Mm-hmm. East African. They had the nice wound, I'll call it, from the tsetse fly. It had been a couple weeks, and they ended up getting treated with the vaccinidazole. Oh. So some interesting. And we only see 1 or 2 of those per year outside of, I'll say, sleeping sickness endemic countries. So.
Yeah, so there were all kinds of violations of this splash pad. They weren't doing things properly. And this was a 16-month-old child.
Oh, gosh.
Oh,
who
terrible.
Died. Because the water, kids having fun, and oh my God. Can you imagine?
No, it's just
it's terrible.
All right. Christina, which of these 2 papers you want to do first?
I suppose we should just go for the meaty one, and then if we have time for something a bit lighter.
Go for it.
Okay, so I have chosen a quite beefy research article to talk about today, which has been published in Science. So the title of the article is Systematic In Vitro Evolution in Plasmodium falciparum Reveals Key Determinants of Drug Resistance. Now, there's a whole load of authors in there, so I'm going to mention the first 3 because they contributed equally, and then the final one. So that is Madeleine Luth, Carla Codines Macias, and Daisy Jen. So they contributed equally to that work. And this comes out, the corresponding author is Elizabeth Winseler. And unfortunately on my PDF, I can see it. So they were in La Jolla, San Diego, at the University of California. And let me check. Oh, there's so many numbers. I think the corresponding author is also in that place. So, yeah.
Yeah, a couple of authors from Columbia University, too.
Oh, yes, I can see that here.
My colleague David Fiddick in the microbiology department. And Anne-Katrin Uleman. Yeah, you must know her. She's an ID person, right?
Yeah, Anne-Katrin, David Fiddick.
Yeah.
Yeah, no, there's a lot of people that... Jennifer Small Saunders, who actually was a fellow. I was one of her mentors. I was her clinic mentor, I guess. Yeah, so there's a lot of familiar folks here.
Okay, so shall we dive into it?
Mm-hmm.
I might need a bit of help now and then.
We're here.
No, that's good. So I thought maybe just kind of as an introduction, maybe just talk a little bit about malaria, actually. Drug resistance. And actually, it's really surprising how quickly resistance to antimalarials develops after a new drug has been introduced. So, for example, chloroquine was introduced in 1934, and in 1957, resistance was reported. For proguanil, the time was much, much shorter. So that was 2 years between the introduction and the first reports of drug resistance being developed. So, this can actually, for the pyrimethamine, the same year of the introduction. So is really a big issue. And actually, drug resistance has been observed to all commonly used antimalarials, including the very newest ones, the artemisinin, where we have seen recent reports of slower clearance of parasites upon treatment. So really, this is an issue for all malarias, so those caused by falciparum, but also vivax and the other parasites. And so when we just kind of look at ACTs, which are to kind of recommend the first-line drugs for P. falciparum infection in most places, actually there's been resistance in 4 countries in Southeast Asia, starting in 2010. So that included Cambodia, Myanmar, Thailand, and Vietnam. And the resistance is apparently usually to the artemisinin part of the drug. So the combination as such generally still works, but I suppose that explains the slower clearance of parasites that has been observed. And then partial artemisinin resistance has also been detected and confirmed on the African continent, for example, in Rwanda, Uganda, Eritrea, Ethiopia, and Tanzania. And then also, I'm just trying to think, the geographic region of Papua New Guinea, I suppose, I don't know, north of Australia. What is that part called? The Pacific region?
Don't know.
Yeah. So anyway, so it is a problem. So any kind of research that is looking at Plasmodium resistance, drug resistance, I suppose, is just going to give us more information on how we can potentially avoid it, or how we can maybe select compounds that are less likely to lead to resistance, or maybe do so more slowly. So at the moment, we kind of investigate drug resistance in patients, so by monitoring the effectiveness of drugs. And this is really done by collecting surveillance data from all around the world. But also we just kind of undertake studies in the lab. So for example, population-based association studies of sequenced isolates that can be done, but also in vitro evolution of resistant parasites. So when we then take a parasite and we expose it to different drugs at different concentrations and just monitor the survival and growth and other phenotypic traits. So that is actually they're both quite time-consuming methods. So that's what we do at the moment. So do you have anything to add to that kind of, I don't know, just kind of warming us up?
Yeah, no, I think that's what they're basically, they're sort of laying out, that there's the 2 different ways. One, if you just, you look for this correspondence, unfortunately, who really knows? There are all these changes. There's an increase in resistance. And then the second approach is you actually, I'm going to call it gain of function, Vincent, just so we get flagged, right? And this is where you actually expose the parasites to a small molecule inhibitor, and you're selecting for resistance. And that's really going to be the bulk of this paper, is comparing those two to these functionally selected variants versus the confusion of what is actually an important change versus what they refer to as the passenger mutations. And it is a little bit upsetting that they tend to have started using mutations to refer to what looks like amino acid changes. So, ouch. But
I suppose they do a bit of both, so maybe we could partially forgive them, because they also look at DNA sequence.
Yeah, so evolved mutations, field and evolved mutations. Yeah. So
I'm actually probably guilty of that too. When I was writing my summary, I was just blindly noting down mutations. So I will try and remember to be accurate. Please do correct me.
But I think a really nice sort of state of where we are now is we can grow, we can culture P. falciparum. So you can culture it, you can do all these different studies. And so really a long way from the days of Traeger, right? The first time you could actually, grow this stuff over there at Rockefeller too. Now you can grow this, you can do all kinds of studies on it. So pretty impressive.
Yeah, as they point out, though, it's a lot of work to grow isolates and then select drug resistance and then sequence them. So they would like to know, are there common mutations that you see associated with resistance? And so that's why they're doing this work.
Yeah, and an army, right? They've got this battalion here, right? So really a lot of authors working on this to get us to this research.
And we have over 200,000 isolates and their genome sequences of Plasmodium falciparum. It's amazing. It's all in the public domain. You can go on PubMed and take those sequences and do what you want with them.
Yeah, so actually, what I also wanted to say that most of that work is done in silico. So most of this work is not wet laboratory work as I used to do when I was still doing research. And that's also something that I find really astonishing and amazing, and actually a bit scary that you can do so much really just with data that already exists and that you can make use that. I suppose that is wonderful, really, that these data is publicly available for people to analyze and use.
Yeah, no, I mean, it's great that we have the resource like Malaria Gen's data set with just all this there. But the thing I'm worried about, right, and this sort of might bring us back to thinking about vaccines, which we may or may not be allowed to research anymore in the U.S., but just they sort of comment on what you were commenting, Christina, is that P. falciparum, really it evolves resistance with relative ease, as we've seen as they demonstrate.
So in fact, what they leverage here is experiments. So there's this drug accelerator consortium, and they have done in vitro assays. They've selected P. falciparum for resistance to 118 inhibitors. So they have the genome sequences. So that's what they analyze in part in this paper. They don't do any as Christina said, they don't do any selections de novo.
Yeah.
Yeah, I think they go back to the lab towards the end of the paper, but we'll be a while before we get there.
Maybe they got lost.
So they have a lot of that sequence data, and there's a lot of information when they compare that to two kind of parental genomes, one that is drug sensitive, which is a Plasmodium falciparum 3DC. So mostly drug-sensitive cell line, and also a P. falciparum multi-drug resistant cell line that has reduced susceptibility to chloroquine, mefloquine, lumefantrine, and pyrimethamine. So really quite extensively resistance. So they use these as comparators. And so with that, they could identify in a whole bunch of genome sequences from in vitro evolved P. falciparum strains. And I think you already mentioned there was 118 compounds. Some of these were licensed antimalarians, and others were drugs that are in the development pipeline. So really a whole range of compounds that they looked at. And they did actually kind of combine them into groups. So 95 groups, just to kind of lump together some that are maybe chemically quite similar.
I found that funny. They have 118 compounds and they put them into 95 groups. It's like almost one group per compound. I don't get that.
It doesn't make,
Maybe there's a typo there. I don't know.
I don't know. Yeah.
I mean, that means most of them are just one drug per group. So I don't understand that.
Yeah. One or two drugs. Yeah.
Yeah. Or maybe, I don't know. Maybe in the supplementary data, they would have explained what their rationale with, but I'm afraid I did not look that up. So, so they had 724 clones, so cell lines. They had data on drug resistance of 448 of these, and they were on average 56 times more resistant compared to the parent line. So really, I think quite significant resistance. Now, obviously, that the resistance that's all the different compounds. So that's not generally or mostly the drugs that we use for treatment. So they aligned the sequence data from those clones with the drug-sensitive cell line, and they looked for single nucleotide variants and insertion deletion variants. And altogether, they found 5,560 single nucleotide variants and insertion deletions in that data set. And that averaged of 17 changed genes that were shared within individual compound chemotype groups. So a compound chemotype group, I interpret that as one of those 95 groups that they decided to work with. So, and of these variants, almost all of them, the vast majority, over 4,000, were in the core genome. So, and that is the genome that excludes regions with repetitive sequences, of which there are many in Plasmodium falciparum, I believe. Although I wouldn't bet my mother's life on it. And each clone contained on average 7.65 mutation relative to its parent, of which an average of 1.2 were missense mutations in the core regions. And I was wondering, they do actually mention a lot of different types of mutations. I'm wondering if it would be worth my while briefly explain what mutations they're talking about. What do you think?
Sure, I can bring us to figure 1A and go through a little bit.
Okay.
So there, people, this is unfortunately not open access. So
I forgot to look that up. I'm sorry. Yeah.
No, that's okay. So for folks that can look at this, it's okay. We'll sort of walk through it. So figure 1, what they're doing is they're looking at compound selected versus the control mutations. And they look at non-coding, and we get pretty significant difference there. The compound selected, you don't see a lot. You see a little over 300. But in the control, we see over 5,000 of these non-coding. But then look at missense. And there, it's sort of interesting how they look at it there. But so they're looking at non-coding, they're looking at missense, they're looking at disruptive in frame, and then sort of the in-del. So this is insertion deletions. And then we have frame shift or nonsense, synonymous. So this is where you change the code, but you still end up with the same amino acid. And then they also have these in-frame insert deletions. So you insert or delete, but you keep the coding in frame. So you're still going to get a protein produced.
But
so the only ones, the only ones that where the number exceeds that in the control are the missense, where you change an amino acid. Right? Mm-hmm. Well, the others seem to be equal or less than the control.
Yeah. So I think the overall kind of take-home message for that first set of data is that the overall compound selected variants were proportionally more likely to be missense or frame shift mutations.
Yep. Mm-hmm. Yeah, so there's a look at the different kinds of nucleotide transitions that you would get in these single nucleotides, A to C, A to G, A to T, C to A, et cetera.
I'm not sure what to make of that, right? I mean,
I was wondering. I've actually got a note there. Is this relevant? I don't quite see it.
No, I mean,
it's statistically significant, but who knows if it's scientifically significant? So
I don't know.
They then looked at the amino acid changes, which I suppose is more relevant because that confers, the amino acid confers the function of a protein, and they did actually see that the distribution of amino acid changes was also distinct in those compound selected variants overall when compared to the control set. And the transitions that they found were more likely to be from small to bulky amino acids, such as phenylalanine and tyrosine. And we can see that, I suppose, in figure 1C. I kind of think we could have just summarized that in text, and I could have spent a lot more time on the following figures.
Yeah, but this is good groundwork, I guess.
It is good groundwork, but I really find it quite hard to get to the bottom of it. And then if we look at figure D, they also then looked at where those mutations are.
Mm-hmm.
And so around half of the kind of core missense mutations in those compound selected data sets were located within a protein domain. So, potentially a part of a protein that has a function. So could potentially be binding a compound, or it could be forming a pore, or it's part. So most of these mutations were in parts of the protein that are probably functionally quite important. And by contrast, less than 10% of the missense control mutations were in recognized protein domains, and
so it means that many of these changes are. I mean, again, these changes are selected with the drugs. They're probably functional. They're not just noise.
Yeah. Sorry,
and I think I think that's the point, right? When you go and you select, you're hopefully gonna get more signal relative to noise. When things just sort of change over time, right? A lot of those changes are not relevant. They're not functional. They're not impacting the resistance.
And I actually just found my note here. So actually also the differences in dose distribution was significant with a really good p-value. And so highlighting that a substantial proportion of those compound selected mutations are likely to play a functional role, as you just said, Daniel, and are unlikely to be the result of a random genetic drift.
But it's very complementary, right? Because when you're selecting, you say, oh, well, that's great, because then you're going to figure out what what changes need to occur. But then you also want to ask that same question: but do those changes actually occur in nature? So you kind of need both, I would say.
And I think we'll get back to that in the final figure. They do kind of compare clinical isolates with those compound selected isolates. So that was kind of single nucleotide variants, but they also looked at copy number variants, because they have copy number variants, so having more copies of a particular gene than maybe in the background, that has been shown to mediate clinically relevant drug resistance phenotypes in malaria parasites. So they compared the sequence series with those of the control strains to see if there are any differences in copy numbers. And they did find over 1,000 potential amplification events. And they found that 271 of the 1724 clones harbored at least one of copy number variants with a really highly significant p-value, so that we can trust the information that they found. And actually, they've included amplification in known drug targets, which I think is always reassuring. So a couple of examples here. We have a protein called PFPI4K, which is a phosphatidyl'inositol 4 kinase, which is crucial for intracellular parasite activities in all stages of the malaria parasite. And that's really a promising target for novel antimalarial drugs. And another one, so that I maybe could mention, is acetyl coenzyme A transporter, and they have found copy number variations in there too, and this could potentially be associated with resistance to chloroquines. So, these copy number variations, they did actually find them in known targets that drugs are targeting. If that makes sense. I think that's quite reassuring.
And I like the way they use the Manhattan plot in this context, right, for copy number variations. So you can see there's a number of chromosomal hotspots for these copy number variants.
And actually, we'll be coming back to some of those hotspots or genes later on. I've tried to kind of give some relevant examples for some of this really quite complicated information that we are trying, or I am trying to confer to you. So another one actually that's probably quite relevant because these copy numbers are important for emerging artemisinin resistance. So there's a hotspot on chromosome 14, which looks on the plot actually it's rather tiny, but is being highlighted as something that maybe we need to keep an eye on, and also similar copy number variants on chromosome 1 and chromosome 9. And they find that these duplication sites often occurred in regions where there's a lot of A and T repeats. The Plasmodium falciparum genome is naturally quite AT rich, and also in repetitive reading. So and that kind of maybe tells us something about the mechanisms of increasing the copy numbers. So by using a microhomology mediated repair as a mechanism for these being created.
What I find interesting is that most of these occur in known genes that are known to confer resistance of some kind to something. So no matter what the drug is, you can have resistance by the same region, the same protein, perhaps even.
Yeah, maybe this is just a selective account. Maybe, that's the ones that they are highlighting in particular.
Yeah, I mean, they said we look for evidence of a known target or resistance amplified. And so they say, PFMDR was amplified in 47 independent events.
Yep.
So clearly it's driving resistance to multiple compounds, right?
Yeah, I was going to say some of it is, kind of shared mechanism, right? Like if you have an efflux pump, then that could work against several. So.
Yep.
Okay, so then they kind of look at whether overrepresented genes affect the compound resistance and culture adaptation in the next set of data. So the next set of data shows that these compound selected variants prove useful in identifying potential targets, which then could potentially be further investigated. And they say that experimental evolution is effective really at associating these compounds with targets or drug resistance genes if they appear at a higher rate than expected by chance. So I'm not actually sure what that by chance rate might be. But when we then look at the 118 compounds that were included in this study, so that showed that 47 had a target or resistant mechanism suggested by overrepresented single nucleotide variations alone, 14 by copy number variants and single nucleotide variants combined, and 15 by copy number variants only. So these are really quite significant mechanisms by which these have been found. So for 18 of these components, there's actually additional experimental data from previous research which identified a resistance gene, and only for 18 compounds could they not identify a resistance gene. So I think, they did find a lot of really quite relevant and useful information, but there's still quite a lot to be done as well. I think that kind of makes the approach quite successful, really. And there's an example maybe. I would like to mention two examples. So, for example, a compound called cipargamin, which is an experimental synthetic antimalarial drug. And for this compound, they identified 12 independent single nucleotide variants in a particular gene, PFATP4, which is a calcium transporting ATPase, so an enzyme that hydrolyzes ATP. And then when I looked this compound up, its target has indeed been identified as PFATP4, with the end result of disrupting sodium homeostasis in the parasite. And actually this compound is active against all erythrocytic stages of P. falciparum and gametocytes. So this is where they kind of identified these nucleotide variants, and then that they could kind of look at previously published information, and actually it then all made sense. So, that I think that is really quite neat. And then there's another example that they give of a compound called cladosporin, which is a fungal compound that is a lead compound for antimalarial drugs. So that's not being used clinically. It's just in the early stages of development. And so this acts on a tRNA synthetase, so it inhibits protein biosynthesis essentially, and it seems to be very, very specific to parasitic protein biosynthesis, which I think will make this quite an interesting compound. And this gene has actually been identified in this study with 3 independent CNV, so copy number variants. And I think that is just 2 examples to illustrate that approach could potentially be really useful. Is there anything else that I needed to mention here?
It's interesting. They point out that they can identify genes that appear across different compounds.
That's right, yes.
Multi-drug resistance.
Multi-drug resistance, yeah. So they found 128 genes that were altered in more than one clone, and 53 of these were identified 3 or more times, so quite significant. And these genes actually include several multi-drug resistance genes that we already known. For example, the Plasmodium falciparum multi-drug resistant protein, PFMDR1, which is a transporter that contributes to drug resistance, including artesonates. And they found that in 19 independently derived single nucleotide variants and also 47 copy number variants. And another example is chloroquine resistance transporter, PFCRT, for which they also find quite a lot of independent variants. But
they do find new gene drug pairs, right? Mm-hmm. It's not just old stuff, as no. We're telling you. They found some new ones as well.
Yeah. So they did. Do you want to tell us more about them, Vincent?
Yeah, there's one called desoxypothylone. It's isolated from myxobacteria, and they found single nucleotide variants in the tubulin beta chain, which supports evidence that the mode of action is like Taxol, which inhibits, which stabilizes microtubules, right? So that's quite interesting.
Yeah, I thought that was really interesting and actually noted that down as an example too.
You also have a copper chelator, neocuproene, and they have with SNV. All resistant clones have at least one SNV in a gene encoding a NADH ubiquinone oxidoreductase. And they go on to talk about why that makes sense. So that's interesting that they can pick up new associations as well. I mean, it's important, right?
Yeah, I think that is really going to bring us forward, and maybe, that might be helpful in deciding what lead components might be useful to take forward. Yeah. If they are identified often, maybe as such mutations. Okay, so I was thinking now of kind of skipping to the part where they kind of talk about the compound selected mutations when they compare them to naturally occurring variants. Would that be okay?
Yeah,
I mean,
it's
there's
good.
a lot of data in between, but I think
no, we don't have to do all of it. Yeah,
maybe in the interest of time. So they were wondering whether compound selected changes, I'm going to try and say changes rather than mutations, so I don't get into too much trouble, if these are different from those that are found in nature. And so, it is really an important question in drug development, is whether natural parasite populations can readily attain resistance because of existing genetic variants, and also because the Plasmodium genome is quite plastic.
So should we look at figure 3 at this point? Maybe yes. Like C, I think figure 3C for me, kind of makes the most sense for looking at this. But
yep. So let me just see what they did first, so that our listeners kind of get an understanding what sequences or what they compared the compound selected cell lines for. So they actually have identified and mentioned another big data set that is available in the public domain, and that includes sequence of over 7,000 P. falciparum isolates that were found in 28 countries, so really globally quite well spread, so very representative. And this showed that actually when they were doing that, this showed that the single nucleotide variants identified in their own data set, comparing it to those clinical isolates, that these were actually found rarely in the natural isolates. So the mutations or the changes in the genome that were generated by selectively growing parasites under pressure were not the same ones that they would naturally found in nature. And they also showed that drug resistance genes are less well conserved in nature. And this is shown in Figure 3, which you wanted to talk to us about, Daniel.
I just, it's, putting this graphically, they have this nice Figure 3 at section C, and it's this condensed version of a network linking genes for the same pair of mutations that they're seeing filtered for the gene pair. So, I guess a lot of this is this issue about this overlap, enriched genes that are mutated in the compound resistance selections.
So what then kind of that they use these data sets also really to explore if those compound selected mutations were in specific protein domains. And I think that takes us then on to actually the next figure. For this, really, they looked at 4 different genes that they had previously identified in Figure 3. So, that were kind of networked. And so, so they looked at these 4, and one of them is called PF-Carl, and then we have PF-MDR1, which I've mentioned earlier, PF-ATP4, which I also mentioned, and then new one, PF-CYTB, that they were looking up in more detail. And mutations in 2 latter one, in 2 of these genes, so the CYTB and PFMDR, they mediate resistance to clinical malarias, whereas the mutations in the other 2 mediate resistance to agents that are in late stages of development. And all these genes had more than 10 mutations in their data set. And they can then they kind of wanted to know where those mutations are and see whether those mutations in their compound selected lines were in the same place as those in the natural lines. So they projected the protein structure and they mapped the mutations. And you can see all the models in the supplementary figure, but here in Figure 4, they actually just show a smaller number of those. So they show, and I think I'm going to focus on MDR1, which is the multi-drug resistance transporter. So they show those, and you can see really quite nicely that that's in Figure 4E, that this MDR is clearly forming a pore. So it's a transport port protein that kind of forms a channel that goes through the membrane. And so they did find that for the field isolates, so the mutations that they find in clinical isolates, the mutations or the changes were really dispersed through the entire protein. And this is indicated in green. So if you have a closer look, and hopefully we can show that there's a lot of wee green spots in that protein model. So for the natural field isolates, the mutations, the changes were throughout that transporter or pore. And whereas the compound selected isolates, they seem to be primarily in the domains that form the channel for this particular transmembrane protein. And there's actually only one single mutation that they found both in the clinical isolates and in those that were generated in the lab. This was found in a field isolate with clinical interest. So, and by that, I interpreted that as one that is probably maybe making it difficult to treat a patient, but I didn't really provide any detail. And actually, the picture is quite similar for the other proteins that they looked at. And maybe we could talk a little bit about what this might mean.
Yeah, I don't know why the compound selected would be so restricted, right?
I don't know. Yeah,
but I think unless the field isolates, they've been drifting a long time, right? So, they sustain lots of changes that aren't relevant. I'm not sure all of these would confer resistance, right?
No, probably not. I would think probably because the ones that they selected in the lab, they were probably under quite a lot of pressure.
Yeah.
Whereas the ones also, it was also no doubt over a much shorter period of time. Whereas the ones in the field, they may have been present for years and years and accumulating these mutations, these changes.
I mean, can you imagine the amount of work to go through each one of these, wild type changes and figure out if it's functionally significant or just, happens?
Well, the thing is, if you make a new compound and you find changes in this protein, you already know there are lots of known changes that confer resistance. So that tells you to focus on that, right?
Yeah. Are these just passenger changes? Because then they could. I mean, you could say, but we always see this. It always corresponds, but it might correspond because it's a passenger in a particular lineage, right?
And then it actually finally went to the lab to do some experiments, which I always like. I find that just...
It was still there. They hadn't shut it down, even though it was at Columbia.
San Diego, I think. Oh, yeah, some of them in Colombia as well. That's right.
I think a lot of this is Gates money, Daniel. So
probably,
they might still have some.
It's pretty immune to what.
Okay, fingers crossed. No, because this is huge work, right? I mean, if we're shutting down some stuff, we need to have some other work to be there for the coming plague.
Yes. Anyway, let's do that final step. So they kind of wanted to compare the effect of those mutations or these changes from the compound selections to that of naturally occurring polymorphisms. So they tested the sensitivity of different parasite lines to a panel of compounds. And I've just highlighted one here, the one that is called PF Carl, because it just kind of rolls off the tongue a little bit easier than the others. So they had 50 clinical isolates that were adapted to cell culture, so they could work with them, and 4 laboratory cells. So those are cell lines that were generated in the lab, and they tested them with 3 compounds, and they measured the IC50, which is the concentration of drug needed to inhibit an enzyme by half. And it is quite interesting to see that the IC50s compared to wild type are quite small in the clinical isolates, but much, much, much higher. So the kind of, I suppose it makes sense really when we look back at what we just talked about 2 minutes ago. The compound selected lines were considerably more resistant, and that was the case in the other genes or in the other protein that they looked at as well. So, and I think that kind of brings me to the end of the results section. So shall we try and bring it all together?
Yeah, what do you do with this?
Yes, that was my big question. So, I don't know. I suppose you could learn. I don't know. You could go back to those, for example, to those variants that they couldn't really find any function. Was that a function? Remember, they had 18 cell lines for which they couldn't really find any known mechanisms or kind of. So, you could look at that. We could learn from that. I don't know. What would you do? What would you what would be the next thing you did if you
I'm
one
always,
experiment to choose? Which one would it be?
I'm always curious when I read a paper, like if my thoughts are going to somehow match up with, what I find in the discussion. One of the things that I think is really a challenge, I mean, it's clearly like a science paper. So you've got all this, lingo, right? So you got PF Carl, and, if you're in the trade, you're like, wow, cyclic amine resistance locus. Wow, look, that came up. And like, the rest of us are like, what? PF Carl? What are you talking about? But I think, like, just as you get into the discussion, one of the things they point out is, as you sort of started, Christina, is that P. falciparum evolves resistance with relative ease. And basically half the compounds that they're looking at appear vulnerable to resistance. So do you say, all right, let's look at the ones that it does not seem so easy to develop resistance? So, early in drug development, do you do some kind of an analysis like this and prioritize, as they say, resistance refractory compounds? I mean, that seemed to make sense to me, and that seems to be something they're discussing. The other maybe is just, boy, the takeaway here is much like tuberculosis, much like HIV, this whole using, a single effective drug or maybe just two drugs might not be enough. Maybe you need to be thinking more of like a TB paradigm, a 3-4 drug regimen. You say, oh, but the 1-2 drug stuff, it works just fine. Well, it worked just fine for so many years. And then we're in this battle. We lost quinine, we lost these other drugs. Do we keep marching forward with the optimism, oh, there'll be another one coming off the bench? Or do we start saying, let's start using multi-drug therapy? Because it's really hard for the parasite to have every single resistance that we be requiring, so that we're not selecting for this.
And we can actually see that in the combination therapies that are already used. So there's slow. There, as far as I'm aware, and I could be wrong, there is no fully artemisinin resistant Plasmodium falciparum out there yet, but there's definitely reduced clearance that has been shown in patients treated, and that could be, the partial resistance to one of those compounds. So maybe having bigger combinations than that would be potentially useful.
Yeah, I guess what I worry about is that, usually you'll start with your artemisinin, and it'll have a particular, mode of action up front. And then you add your second drug for sort of that second phase killing, right? So you start your artemisinin, and then you add, Clinda or Doxy or whatever, or, whatever your combo. But maybe we need two sort of quick acting upfront agents, so that you're not, because you're still selecting for the artemisinin resistance. And so someone who's particularly ill, early days, they came in 25% parasitemia. By the next morning, it was less than 1. Now it's not less than 1 till 2 days later, right? So maybe we need to sort of make sure that we're hitting upfront with 2 drugs that have that quick killing, and then,
So I think what we have is a big bucket with really useful information. And I think probably over the next few years, hopefully more research will be undertaken to kind of, pull out smaller elements out of the bucket and try and make sense and maybe apply that.
That's in fact what they say. These results provide a starting collection for algorithms that can be used to identify the mutations that are associated with drug resistance, not just in falciparum, but maybe the other species.
Maybe the others as well. Yep, right? That's right.
Yeah. Maybe also a push for more vaccine, right? Because, right now, right, we had some autochthonous cases of malaria in the U.S. Wait till we have autochthonous drug-resistant malaria cases in the U.S. And with our current approach to the world, right, if malaria runs rampant, more and more opportunities for replication, for selection for resistance, more cases of malaria that are potentially going to spread into areas that currently have not had much autochthonous transmission. So,
Yeah, particularly with a changing climate, when the mosquitoes will move, and also with increased transport. For example, the Anopheles stevensi, which has kind of moved its way from the kind of Arabian Peninsula into essentially the entire African continent. So it was first kind of seen in Eastern Africa, but now we've actually found it in West Africa as well. So
that's a disaster. I mean, so Stephen's eye, right, is moving it into an urban environment. And when a lot of people travel for business and other things, just going to be here for a few days. I'm going to be in the city, not a problem. But when you're starting to see Stephen's eye spreading in urban environments, people flying in and out, it really creates this opportunity to spread. And then couple that with resistance, couple that with poor monitoring, dare I say. If you don't do a test, it'll never be positive until the person ends up really sick in the hospital. So,
and of course, we also have tests that are no longer working efficiently in many parts of the world. Some of these
the rapid
diagnostic tests. So if you add that, if you throw that in, I think we need to throw a lot more at kind of a multi-pronged approach of tackling malaria. And this paper maybe gives us one avenue to explore.
So if you're shuffling around funds, this group could use more funds because this is, I think, critical work.
Yes, absolutely. Yeah. Okay, shall we move on? Do we have time for something a bit lighter instead of a hero, or do you need to go?
You have 5 minutes. Can
5
you do
minutes.
it?
Okay, yes, I can actually.
Okay.
So this is a very short paper, and the title is Paleoparasitological Analysis of a 15th, 16th century latrine from the merchant quarter of Bruges in Belgium, evidence for local and exotic parasite infections, and it's published in Parasitology and is open access. And so I find that a really quite interesting subject, paleoparasitology, and it's a bit of a change from the basic research paper that we just did. And I suppose it's important to keep our mind agile. And this kind of study, so looking at parasitic infections in the past, they've kind of made an important contribution to our understanding of the past epidemiology of parasites and infection in past populations and lifestyle in the past. And in some cases, these ancient parasites can also provide evidence for long distance travel or migration of people in the past. And this is really what that paper is about. So let's think about schistosomiasis, which is primarily a disease of what is sometimes considered the old world, i.e. not the Americas and the Caribbean. And of these species that cause human disease, only one is found in the Americas, so that's Schistosoma mansoni. So how did you get there? And why not the other species? So maybe this paper has a bit of an answer there. So we already know from kind of genetic analysis that this introduction of Schistosoma mansoni, which has been around in the African continent forever, is really a relatively recent introduction in South America from Africa. And the proposed timing of this introduction coincides possibly with the Atlantic slave trade in the 16th century. And the study here takes us to Bruges, which was a very important commercial hub in the medieval period. And so really the economic success was built on import and export of goods, for example, fabric. And so they had kind of, that led to the development of commerce and banking systems in Bruges until it was kind of superseded by Antwerp, which is another big system. But Bruges was the home from merchants from Spain and Portugal and Italy. And we know they traveled to the Americas extensively, I suppose, to rob the local peoples of their gold and bring in diseases, although that may be less deliberate, that latter part. And actually, they established nation houses in Bruges, which function as administrative hub. And the study in this paper actually focuses on a particular latrine of a Spanish nation house. And they kind of also use data from previous studies in medieval Belgium, which kind of gives us an understanding of locally transmitted parasites. So, if they find something that shouldn't be there, they'll find it. So they analyzed multiple layers in that that latrine, with the lowest mostly consisting of fecal and other organic matter, and the top layer was kind of the final fill before that was abandoned. And they analysed 3 samples, which I'm not going to go through the details of that, but you can read it, and it's really quite readable. And they recovered a huge number of parasite ova. So they found Ascaris, they found Dicrocelium dendriticum, which is a liver fluke. They found Tenia, perkworms. They found Trichuris, and I think Trichuris are really the most abundant.
Amazing
And
how well preserved the eggs are, right? I mean,
very well preserved. So there's some beautiful
yeah.
pictures in there, yeah. And they also potentially found some fasciola or maybe an echinostoma, so an intestinal fluke. They're difficult to tell apart, those eggs, really, particularly if they're maybe a bit aged. The one really interesting thing is what they found is a Schistosoma mansoni egg, a single egg. And that shouldn't be there. That wasn't a local parasite. And actually a Schistosoma mansoni egg has only recovered in one other place in medieval Europe, I think from a latrine in France. So this is exceedingly rare. And so really that kind of provides evidence probably that parasites of migration occurs with migration of humans. So if it was no doubt, one of those Spanish merchants who went to do trade in Africa, brought the schistosome back home and then took it over to the South Americas. And it was maybe not that guy who pooped in this particular latrine, but potentially, it was a similar pathway. And actually, that kind of also makes sense with the biology of the parasites. So in South America, we actually find the genus Malaria, which is the snail intermediate hosts. So in South America, if eggs were shed, the life cycle could be completed. The adults also, they live for quite a long time, so 3 to 5 years, but can be potentially longer. So they would actually survive long enough for someone to cross the Atlantic to get into the Americas.
Easily.
So, and I think it's quite an interesting thought experiment, really, to consider that, not just things like smallpox were imported, but also parasites, and as a result, really, of forced movement of people from Africa to the Americas with the Atlantic slave trade. So this infection may have been in a merchant who acquired a parasite during traveling in Africa and then going to Bruges and eventually ending up in the Americas, because in Bruges, the parasite would not have been able to complete the life cycle. So, because we don't have the right genus of snail.
And he was dropping the eggs in the latrine, which is not conducive. So
that's great. Thank you, Christina.
You're welcome. I just thought something slightly lighter hearted.
That's very cool.
Yeah, good.
All right, that's TWiP 256. Show notes, microbe.tv/TWiP. You can send comments, questions if you're doing a clinical guest. TWiP at microbe.tv. Please support us, microbe.tv/contribute. Daniel Griffin, Columbia University, Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Everyone be safe.
Christina Nall is at Glasgow University. Thank you, Christina.
You're very welcome. I'm all talked out, so I'm looking forward to a glass of wine.
Well, it's late there, so you're all set.
Yeah, it is. Yep.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP is parasitic.